Thank you, Adriano, for joining us today. Well, the first question for me is basically, you spend most of your career studying prions, right? And I was wondering if you could tell us in simple words what prions are and why they are so dangerous for the human brain.
So, when I started working on this, which was around 35 years ago, very little was known about prions, and most people thought that, there were these diseases, but most people thought that there had been a virus, that there has to be a virus somewhere, and then it turned out that there was no virus, nobody was able to find a virus, and then it was Stan Prusiner in San Francisco who came up with the idea that maybe it's not a virus, maybe it's just a protein that misfolds and replicates, and that turned out to be true. Yeah.
Wow. What is dangerous? What does it do exactly?
We don't know. I mean, that is really one of the focus in my lab. And it's a bit, I would say, almost embarrassing that we still don't know exactly how it damages the brain. But at the same time, I would argue that's true of most neurodegenerative diseases. I mean, after all, we also don't understand how A-beta and tau damage the brain in Alzheimer's disease or synuclein in Parkinson's disease. So, neurodegeneration is still pretty much of a black box.
So in that sense, considering that it is indeed like one of the biggest challenges that comes with aging, what would you think is the most dire question that we need to answer when it comes to neurodegenerative disease?
I would reframe the question a bit differently, because it's. I think that. In general, of course, we need some therapies, but that is a very generic statement. Of course, we need therapy for everything, I think that we need to better understand the pathogenesis and really the molecular steps that generate the disease. And once we have understood that, then it's a time to develop therapies. My feel is that there have been a lot of false starts in neurodegenerations. Most of the therapeutics that have been developed have not really worked, and I think that the reason is that we need more basic sciences, but because we still do not really quite understand what are the targets, the molecular targets that should be hit. And if you don't have that, there is not much you can do, because then you're a bit lost. Yeah.
And I think for that your lab, is developed cutting-edge 3D technologies, imaging technologies for that. So what could be it useful to give us, what new inside these maps could be helpful into the human brain disease?
Yeah, the 3D histology, I think, is something that we put a lot of effort into, and I think that for me, the reason was really to try and get a holistic representation of all cells in the brain in a rapid, quantifiable and natively digital manner. And because, the way, you can do morphometry, you can take brains and make step sections like every 100 microns, and then you can digitize them essentially with a microscope, with a camera or scanning microscope, but all of this is extremely laborious, and you still only get every 100 microns a picture, so you don't really get the whole thing. And so for me, light sheet microscopy, and that's essentially what we did. We spent a lot of time developing methods to rapidly clear brain tissue and then acquire it by light sheet microscopy. And I think that the one advantage is that that optical sections are much less laborious than physical sections. Essentially, an optical section can be done in a fraction of a second, and the physical section takes hours. And so that works very well, but of course then it's not only about clarification of the brain and acquisition of the images. Then you have these huge files, terabytes of data that need to be processed, and the size of the files is such that it's even a challenge to transfer them from one computer to another. It's still there are lots of bottlenecks, and of course then you have to extract information from that. So it's a challenge. It's I would say at the limit of what reasonably priced computing can do nowadays, but we're getting there.
It kind of replaced the manual bottleneck, that we had before.
Yeah, because and the manual bottleneck is really there because we run an institute of pathology which makes around 270,000 histology sections a year, and essentially everybody who gets surgery will have some kind of histology on anything that is taken from the body, and we have an army of 35 histology technicians who spend the entire day making sections. First of all, it's a relatively boring job, but it's also a very specialized job. That is, you need actually people who know how to do this and who are very well trained, and so it's also very expensive. So there is a lot of motivation to get away from this type of procedures.
So the trade-off is worth it, despite the challenge and the storage challenges and everything. You believe that this is important, the trade-off between manual and
absolutely. No, I think that in the end you could argue that histology has not changed for the past 200 years. It's the way we are examining tissue has never changed, and so it's time perhaps to rethink the way we look at tissue.
Okay.
And you do say that there are a lot of unknowns and a lot of things that we haven't uncovered, but I think it is very apparent that the research that you have done has had also tremendous influence on public health decision making, right?
Yeah.
So in this sense, how important would you say that the basic research is in disease prevention? Should it be given more importance than in the current society, or?
Yes, but one has to be careful because I think it's a bit too obvious to say, yes, we need more research. Of course, if you are a researcher, you will always say, we need more money, we need more research. But I think that's not enough. Because to some extent it's self-serving. So I think one has to argue in a more convincing manner. And I would say that the argument in favor of doing basic research to understand and treat diseases is actually a historical one. For example, something that one can, which I like to point out, is in the 1970s. Richard Nixon, the American president, declared the so-called war on cancer. So the idea was in 10 years we will defeat cancer, and it didn't happen. Now we have a lot of progress, but at that time, until 1980, between 1970 and 1980, it certainly didn't happen. But then other things happened, because a lot of money was injected into basic research, and then, Howard Temin and David Baltimore discovered reverse transcriptase, which was a very weird enzyme that appeared to play a role in a viral infection of mice called Maloney-Murine leukemia virus, which was of zero relevance to anything human, so you could argue that this was completely irrelevant research, and I mean, it received a Nobel Prize because it was a very novel. The idea that you can make DNA from RNA was a complete paradigm shift, but there was no practical application. And then only 3 years later, AIDS came about, and then the only reason why we then could develop diagnostics and therapies was because there was an understanding of what is a retrovirus, and and there are many such examples, but I think that this shows you that it's good to say, okay, here there is a disease, can be Alzheimer's, diabetes, whatever, and now let's do translational therapy aimed at finding a cure. Problem is that works if you know exactly what causes the disease, and then it's very powerful. I would argue it's not necessarily something that academia is very good at doing, but industry is very good and can do that. But if you don't know exactly how the disease works and what is the cause, then you have to step back. You can't do the second step before the first, and that's why the other thing is that basic research is a bit of a lottery. Many things don't work, but also in a lottery, the more tickets you buy, the more likely it is that you will win. Yeah. So there is both.
You were saying like you didn't come to Italy since 50 years ago, and I'm wondering what Italy could do better to improve, motivate scientific research and retain talents like you in Italy.
I mean, I think that Italy has improved a lot. When I left Italy in 1978, and I sporadically come back. I'm involved in a couple of things. I serve on the board of the Italian Institute of Technology since many years, since 15 years, and I have seen the IIT grow, and I think it was really a very good example of how you can actually do stuff in Italy, and that was at a time when I had already lost hope that anything could be done, and but IIT showed that this was possible, and then the Human Technopole was created, and it was a very similar idea. So the idea was really that the Technopole would be a bit like a sister organization to the IIT, and that is working also very well. So there is a possibility to do stuff in Italy. And now to the second part of your question, how to retain the Italians in Italy, I think that we should not. I think you should not retain the Italian scientists in Italy. But I think that the more important thing is there is a global pool of talented people, and they are everywhere in the world. And the question is, how can Italy tap into this global pool? Some of these talented people will be Italians, others will be Germans, Americans, Brazilians, whatever, and then the question is why would these people prefer Italy over other places? And I hope it will not be the Bella Vita, but it will be actually the scientific opportunity to do some great science, and that's I think what the aspiration should be. And yes, I mean, getting back Italian talent from abroad, I think it should be. But if you can't get Italian talent, then get the Chinese talent. I mean, I don't really see this as a contradiction. Yeah.
I see.
I feel like in most of your replies, it's coming out how still excited you are about the research that you do, right? And in general about the questions that we have. So after having such a long and fruitful career that is still ongoing, right? What still motivates you? Like, what would you say is the biggest drive to still persevere in research?
That's an interesting question. I think that that is often misunderstood. I was a journalist, a Swiss tabloid journalist, wrote about me I think it was a very bad article, and here, Aguzzi has reached everything he wanted in his life. Complete misconception. It just shows that that this guy has no idea of how I function. I mean, for me, the goal of improving my career, getting higher, and whatever, it has never been. I mean, what I like is to hang out with a bunch of students and look at Western blots and plan an experiment, and it gets never boring because it's always something else, and and that is very satisfying. And I mean, it has. I have never felt that I would need anything else. So I also, I mean, the problem with, I mean, some of my research was very successful. Other things were not so successful. But the problem is when you do something successful, then everybody comes to you and tells you, do you want to be the director of this, the president of that? I never wanted that, and in fact, I actually pretty much actively sabotaged this because all I wanted was to run a lab, yeah.
Very inspiring. Really.
No, well, I mean, I think in a way, if I think of Marino Serial, I think what he does here, running this big institute, is very altruistic because he is like me. I mean, he likes to do science, and so I think that it takes some generosity to step back and say, look, I mean, I know how to run a big institute, so that's what I'm going to do. Although actually doing experiments is more fun. But I am not that altruistic. I mean, I prefer to do what I like to do.
I see. So, at the end, what for the young scientists that are like interested in or beginning their career in neuroscience and pathology, and they are kind of interested in, especially like you, in high-risk disease and long-term research. What kind of advice you would give them?
Well, I think each one of us is different and has different goals and dreams for their lives, and science can be ultra satisfying and exciting and very fulfilling and can give a meaning to your life. But there are many other professions that you can do as a biologist or as a biomedical researcher that are not academic science but are equally satisfying. And back in my days it was more difficult, but nowadays you can do so many different things. You can go into academia, you can go into a non-academic research institute like this one, and which is not a university, but it's a kind of a basic research institute. You can go into biotech. Biotech companies nowadays, many of them do research which is just as innovative and exciting as the research that you would do in an academic setting. You can go into pharma, you can even go into government and advise the government about policies, and some scientists were, including a Nobel Prize winner, were extremely successful and impactful at that. So there are many different avenues that can take, and the important thing is to note that there is no hierarchy. It's not like becoming a tenure professor is the best thing you can achieve in your life and everything else is less than that. They are just different, but they are not, it's not like one thing is higher or better than the other. And I think that I think that also with respect to starting a PhD, I think that what I see sometimes is that young scientists don't really understand exactly what is a PhD, what it takes, and whether it's actually the perfect fit for their career. And perhaps, that can be tragic, because then sometimes people start doing something that is not really going to help them a lot, and then a lot of frustration arises, and so I think as mentors we should also try to avoid these situations. Yeah.
I see. Well, I mean, there's one question I like also ask a long-term researcher like you and like successful one: work-life balance.
Yeah.
How have you like through the whole research career, were you able to maintain a how was it for you? Like,
well, I think that work-life balance in some way is absolutely crucial, and in fact, I think that sometimes I really go to some people in my lab and tell them, look, go home, because you're working too hard, and and I know that this is going to end up with a burnout, because if the only content of your life is your research, the problem is that research is very frustrating because many things just don't work, and but if that's the only thing that you have in your life and it doesn't work, that's a complete disaster, and so you need to have a balance. You need to have something else in your life, which could be sports or family, whatever. I mean, but you can't only work, and because your work will suffer. And so yes, work-life balance is very important. Now, the thing is, I personally have never cared for work-life balance. I hardly ever took any vacation in the last 30 years. So what I can say is, do as I say, don't do as I do.
Cool.
Thank you so much.
Thank you very much. That was very nice.